chromogenic medium CCA, chromogenic coliform agar, is a selective culture medium for the detection and enumeration of coliforms and E. coli with an adjusted and precise selective capability ideal for water analysis. With antibiotics cephalosporin and vancomycin included in its formulation. Improved results and recoveries. Compatible with a variety of different membrane brands, using our combination of CCA and coliform CV selective supplement. Formulated according to an alternative method proposed by the Spanish Ministry of Health. Escherichia coli, E. coli, is a type of coliform, which has been selected as an indicator for the presence of coliforms, other enteric pathogens, and direct or indirect fecal contamination in water and foodstuffs. CCA is a solid, selective and differential culture medium used for the detection, enumeration and isolation of coliforms and E. coli in water samples by the membrane filtration technique, in particular in water for human consumption. Formulated according to an alternative method proposed by the Spanish Ministry of Health and Consumption, without prejudice to the method laid out in the European Directive 98/48/CE. Method validated for the microbiological analysis of coliform and Escherichia coli, according to the method of reference ISO 9308 by Culture and Chapman TTC agar. The colonial differentiation is provided by the chromogenic mixture, composed of two enzyme substrates. SAM and GAL for the determination of the beta-D, galactosidase enzymatic activity. X-glucuronide for the determination of the beta-D, glucuronidase enzymatic activity. All coliforms have beta-D, galactosidase activity. E. coli has additional beta-D, glucuronidase activity, unlike other members of the coliform group. SAM and GAL forms a colored precipitate in the presence of beta-D, galactosidase and gives a salmon pink to red color for the coliform colonies. XGLUC forms a colored precipitate in the presence of beta-D, glucuronidase and turns the E. coli colonies a blue color. However, the presence of the two substrates and cleavage of these by E. coli results in the formation of dark blue to violet colonies. This medium has an adjusted selective capability, which is very effective due to the antibiotics mixture included in its formulation. This selectivity is the result of presence of cephalosporin, which inhibits Pseudomonas and other gram-negative oxidase-positive bacteria, and also because of the presence of vancomycin, which inhibits the growth of enterococci and other gram-positive bacteria without affecting the recovery of coliforms. If you use our new sachets, you can pour the powder directly into 500 milliliters of distilled water without requiring previous weighing. Weight of standard 500 grams bottle, you have to suspend 29,45 grams of powder in 1 liter of distilled water and bring to the boil. Do not autoclave. Do not overheat. Cool down to 45-50 degrees Celsius and add the content of 1 vial of coliform CV selective supplement for coliforms to every 500 milliliters of medium, ART. Number 6140 LYO. Homogenize and distribute into Petri dishes. The use of the membrane filtration technique is highly recommended. The water sample is filtered through a membrane filter of 0.45 micron pore diameter, validated according to the ISO standard 7704, 1985. Use standard equipment for filtration. With the help of sterile forceps, place a 0.45 micron filter on the filtration apparatus. Hold the grid pattern upward. Take 100 milliliters of sample for each test and vacuum filtrate it. Again, with the help of forceps, remove the filter and place it on the plate, holding it in the same position and avoiding the formation of air bubbles so that there is complete contact between the filter and the plate. Proceed to the incubation of plates. Incubate the Petri dish with the membrane at 36.2 degrees Celsius for 18-24 hours. If after 18 hours red or colorless colonies are observed, extend the incubation to 24 hours to include late reactions of beta-galactosidase or beta-glucuronidase. Enumerate colonies as described. E. coli, deep blue to violet colonies. Coliforms, salmon pink to red colonies. Other gram-negatives, mainly enterobacteria, colorless. Total coliform is obtained by counting the salmon rose to red colonies and the deep blue to violet colonies. Calculate the concentration of coliform bacteria and E. coli in 100 ml from the initial volume of water filtered, and the number of characteristic colonies counted on the membrane. The results are expressed as colony, forming units per milliliter. Confirmation of E. coli presence is required in Dole test. To confirm the presence of E. coli colonies in this medium, a small amount of tryptophan is added. To verify the production of indole, coat the blue-violet colonies with a drop of Kovacs reagent, art number 618. If the reagent turns to a cherry red color in a few seconds, positive production of indole confirms the presence of E. coli. Traditional methods for E. coli in water samples are tedious because a lot of different colonies have to be identified. These methods present different difficulties for the microbiologist. For instance, the reference method ISO 9308, which is based on culture in Chapman TTC, European Directive 9848 SA. Some media allow coliform detection and enumeration in 24 hours, but they do not differentiate E. coli from total coliforms. The chromogenic medium allows identification of E. coli directly, as the colony has a different color than the rest of the flora, allowing a direct and simple enumeration. Advantages of chromogenic media: better diagnosis, positive identification of E. coli, detection of E. coli, total coliforms, and other enterobacteria at the same time. Direct identification and enumeration of E. coli and coliforms in 24 hours, which saves time. Easy interpretation of results. Simple and easy method. Advantages of CCA. High recovery rates with different brands of filtration membranes, especially when using our coliform CV selective supplement. Our CCA agar is the ideal medium for detection and differentiation of coliforms and E. coli in water. The presence of Tergital-7 and the addition of antibiotics cephalosporin and vancomycin, coliform CV selective supplement, reduces the growth of accompanying flora, resulting in excellent recovery rates and specificity for coliforms and E. coli. Also ideal for wastewater. With the new selective supplement, coliform CV, our CCA agar is even more selective, ideal for the analysis of wastewater and very contaminated samples. Better color contrast for easier interpretation.